I have to say this, it's an honor to be here. I'm really thankful for this opportunity to show my work and maybe to work with you in the future in the Institute. I will be talking on a topic that I have been working in the last 10 years. It's non-equilibrium thermodynamics of active matter. Currently, I'm at the University of Ljubljana in Slovenia. First at all, I want to mention some of my collaborators, colleagues, and advisors in Colombia, Professor Daniel Barragan at the University of Colombia, my PhD advisor in Barcelona, Professor Miguel Rubí, in the University of Barcelona, and then my supervisor in Norway in the Pore Lab, Professor Signe Gjelstrup. Right now, I'm working with Professor Giga Kos in the University of Ljubljana and also in the Stefan Institute. And right now, I also have a couple of projects with Ignacio Borde out at the University of Chile. So my presentation today is divided in 5 sections. I will just give a brief introduction. First, I will talk about non-equilibrium self-assembly. That is the simplest version of self-organization in active living systems. Then I will talk about active particles that can be synthetic and can be biological. Active kinematics that, for instance, my current project is in microtubules, in the system of microtubules with these myosin molecular motors that allows them to move and to create these pneumatic defects that I talk a bit later. And finally, one of the most interesting projects for me is self-sorting, to understand the sorting of the cells that is related with morphogenesis of several tissues and system of interests. So the introduction is based on a paper, a review that I wrote in 2019. So as far as we know, active matter is inherently out of equilibrium. It means there is irreversibilities, energy dissipation. That dissipation takes place at the level of the discrete constituent or discrete objects in the system, taking this ambient energy to convert into mechanical work. I say mechanical work because it could be another kinds of works. These systems, or in active matter, will also find self-organization. That, of course, is not going. We cannot describe this with the classical statistical mechanics. I'd like to separate the active matter systems in 2, the synthetic and the biological ones. I have been mainly working in this interface between the chemically and the biological one, working with Janus, catalytic particles, active liposomes, microtubule motors, complexes, and tissues. Then I want to introduce about the dissipative structures. This has been a topic that has been, we kind of have some solid ground to understand, and it's pretty similar to what we define as active matter, just that there are not clearly discrete constituents in the systems, and typical examples are the Bénard cells, Belousov-Jabotinsky reactions. We have technical problems. Maybe I will just put it a bit closer. Hopefully, it's going to help. I will stay also closer to the computer. So these Belousov-Jabotinsky reactions, they were studied also by Prigogine, Nobel Prize. These are spatiotemporal patterns. We can describe it through simple partial differential equations. And then, for instance, hurricanes, that is something most complicated in the macro scale, atmospheric scale. This is a dissipative structure. It's a clear example in nature. So just to mention key insights here, all active matter systems are dissipative structures, but not the opposite. Not all dissipative structures are active matter. The key is in this unit, in the discrete unit. So since we really understand and currently in active matter, we are aware that dissipation is a key concept, a key variable to understand the systems. We are in the field of non-equilibrium. So, and there are different kinds of non-equilibrium. I will try not to talk a lot about this part here, but I prefer to differentiate the types of non-equilibrium defined by the bath, because if we have a bath at equilibrium, we just have the relaxation processes where detail balance is fulfilled, the global detail balance, and then is the Gibbs formalism, and we can describe processes as the glass transition, nucleation, the classical nucleation, some crystallization, and so on. Then, when we have multiple baths that can be at equilibrium or close to equilibrium, we will have, if the system is open, we have the steady state system that fulfills is the local detail balance. And in this kind of systems, we can have some synthetic active matter like the catalytic Janus particles. No, sorry, the catalytic Janus particles, no, these particles, the Janus particles that exhibit thermophoresis or diffusion phoresis that can be described very nicely through linear non-equilibrium thermodynamics, the field that developed on Sager and Peter Masur then. And then we have the other kind is when we have the baths out of equilibrium. So in active matter, in most of the system of active matter, we are in this case where essentially baths cannot be assured that are in equilibrium, but maybe they are close to equilibrium, and we can define locally the local detail balance in the correct or in the appropriate level of description. And this is where we find most of the self-organized active matter and living matter systems. And we can describe this with the mesoscopic non-equilibrium thermodynamics. That is a formalism that I'm going to talk more tomorrow. And then there is a particular case that is when you have a system that is only composed of particles, that they are active. Hopefully they are going to fix. Sound. Hilda. Should I continue? This specific case where we cannot define baths because the bath itself are the particles, then I agree this is a system that is completely out of equilibrium, that breaks all detail balance and all possible restrictions. And I call that like dry active matter. And then if we cannot define locally functions, then I don't know if we can do thermodynamics. So it's a question mark. So let's continue. Why thermodynamics? Why I keep saying thermodynamics to describe this active matter system? And it's because it is not going to replace the mechanistic interpretation or the models, but it will help to organize and constrain it. Why to constrain it? Because we have the second law. We know that the models and the interpretation of the results should fulfill the increment of the entropy. So the entropy production should be always positive. It will be useful when we are going to do coarse graining, because usually when we go from the microscopic scale to the macro scale, we are losing information, or we are obtaining equations that maybe aren't going to fulfill the second law. When doing it through thermodynamics, we assure that we are going to keep that. We can also propose predictive criteria based on the energy dissipation. That is the key variable in these dissipative systems. And it clarifies the energetic origin and cost of the processes. And the best is that thermodynamics allows us to connect different fields, different areas, because we can talk about chemical processes, physical chemical processes, physical processes, and biological processes. So that's the reason I work with thermodynamics. So The research question that I proposed back in the time in my master was something like, if can we build a general non-equilibrium framework based on entropy production to explain self-organization in active systems? So we are still working on that. We already have some good results. But then the idea is that with thermodynamics, we not only explain how, but also why self-organized structures emerge. So the how is more the dynamics. The why is matter of the energetics. And the point is that understanding biological organization, we can maybe reveal some principles underlying the origin and evolution of life. So that's the big goal. Okay, now let's go to the second part that is about non-equilibrium self-assembly. As I said before, it's a previous step to understand self-organization. Why? Because in self-assembly, we don't have these complex feedback loops that goes almost into the field of complex systems. And the idea is to find the relationship between the structure formation and the shape of the structure or the morphology with the energy dissipation to understand the mechanisms behind self-organization. And for that, I work with mesoscopic non-equilibrium thermodynamics, because the main thing is, okay, let's do thermodynamics, and this is the heart of the formalism. This is the Gibbs entropy. It does not necessarily depend only in the trajectories as currently done in stochastic thermodynamics and another fields of non-equilibrium statistical physics. So the Gibbs entropy, the differential depends on this is called the chemical potential and the variation of the probability in the space, in the generalized space or in the mesoscopic space. And from this, we can obtain the entropy production, and from the entropy production and on Sager's formalism, we can obtain the Fokker-Planck equations or Langevin equations, the macroscopic equations, so we can obtain the dynamics, and when obtaining the dynamics, we are able to compute the important variables to obtain the entropy production. So that's the key point. From entropy production, we obtain dynamics. Once we have dynamics, we estimate or compute the entropy production that times the average temperature, we will have the energy dissipated or the loss-free energy. So during my PhD, I proposed a criterion for the formation of these self-assembled structures out of equilibrium. So the key idea is that, of course, if we quickly, from an initial state, we can have different structures that, like I'm illustrating here, but we are going to observe that with different proportions, different probabilities, and of course it's going to depend on the trajectories, but not only in the trajectories, but also in the constraints and in the nature of the building blocks that are the discrete entities. And for that, we propose an effective potential that, since the system is dissipative, we should consider this, that is the total entropy produced in the process, and this is other term that accounts for the energetic cost to do changes in the shape in the structure. So I'm talking very quickly. This first part, I will go fast. We can also describe the probability. If you see, it's pretty similar to the Gibbs probability, but this is not the free energy or Helmholtz free energy. This is an effective potential.
Say what eta is?
Sorry?
Eta.
Yes, so eta is the parameter, is the parameter characterizing the structures or the shape. So for instance, if for eta 0, we have a disk. For eta equal to 1, we have the sphere. So in this case, eta can be seen as the sphericity, but not necessarily. But the key point, this eta is a variable, it's a structural variable characterizing the structures or the morphology.
I understand that, but I don't understand how you parameterize eta. Why do you call 0.3 a cylinder and 0.5 a...
I don't know, sorry, this is just for illustration. This is just for illustration.
But I think it's important, because if you don't know what eta is, I don't know what this integral is.
No, you are right. No, you're right. In every case, so indeed, the most important on this formalism is to define in the best way what is going to be your structural parameter. In this case, for instance, if we define a sphericity, if we are just in this region, we can define a sphericity from 0 to 1. If I want to characterize the structure with a
I'm not asking about how you, I'm just asking how you parameterize it, because it seems you need a measure that tells you which ethers, I mean, the way you're defining it now, there's no measure for ether, right? All ethers are equally weighted, right?
Yeah, for instance, this, how to try to respond that, it's, if you see the model is a parameter that goes into the model. We don't compute eta. Eta is a parameter.
But if you reparameterize, does it change the formula, or is the formula invariant under how you reparameterize eta?
Yeah, no, it does not depend on how you parameterize. Okay. No, yeah. So this eta could be whatever, sphericity, size. I'm going to illustrate a bit more. For instance, one of the studies cases was in the formation of Lysian rings. Quickly they are formed because of the diffusion, reaction, and precipitation of salts. If we solve the partial differential equations, we can have that. This is the typical reaction diffusion system. But the point is that the distance between the rings, if we have a 2D system, they are going to form rings. I'm just showing half of the system. So the distance between the rings and the variation of the size of the crystals that precipitates can be characterized by a unique parameter that is this eta. This eta, I have not here the values, but it can vary from 0 to 1,000. So whatever. The point is that it parameterizes the different morphologies that we can observe in the structures. So in the model, in the theory, we can have all of these systems, but experimentally, we only observe one, at least from the experiments we had access, and it's for this configuration. And of course, this when we solve all the dynamics and compute all the free energy chains and the dissipation, the point is that this structure is not found for the minimum of the free energy, or the classical free energy, reversible chains of the free energy, but is found to be in the minimum of the proposed effective potential that is close to the minimum of the entropy production rate. Sorry, the total entropy production that is here. I'm not putting the values, but this in the model is a parameter that helps us to understand what is the morphology of the pattern we are having. If we go to another study case, that is the magnetohydrodynamic structures. So quickly, more or less, is that you have an interface between it's air and a fluid. You set in the interface magnetic particles, and behind the system rotating magnetic field. The particles would have this for 10 particles, we have 2 possible configurations. For 12 particles, also 2 configurations. That is, the difference is the number of central particles. So here eta is going to be discrete and takes only 2 possible values. So it's a pretty easy system. And also to compute the dissipation is easy because it's just the viscous dissipation because of the fluid viscosity. The point is that the points are the experimental and simulation results, and the dashed lines are the results for our model, our criterion. This is a work done by Tetrakov and Grybowski, both Polish people working in the United States. And the point was to prove that that we were able to see that. For instance, here the most likely structure for a given Reynolds, in this case, is the one that dissipates more energy, opposite to the previous case of the Lysian rings. So it's not the matter of if it's a minimum or a maximum in the entropy production, is that they are the minimum, the most likely structures are in the minimum of this energy potential, effective potential that we built. So we studied also the energy dissipation in cancer tissues. We wanted to understand the transitions between the different stages of cancer, specifically this cancer in the liver in rats. This work was mainly done when I was in Norway. So basically, and we propose a very simple model. So basically, the tissue is a porous media where we can have flows, diffusive flows, where we can have chemical reactions. And if we set an external force, as for instance, this ultrasound that is now they are studying the ultrasound as a treatment to improve the transfer of medicaments in the tissues. So that's the reason I started working on that. We can compute what is the energy dissipation for a given state. And from that, we can build like a map to be able to compute our effective potential. So basically, we can characterize the tissues in a very simple macroscopic way with the porosity and the Young modulus. Yes.
This is the energy dissipation for the variables you're keeping track of.
That we can keep track on, because there are two.
I guess I was going to ask, at an order of magnitude, how does the energy dissipation that you're computing compare with the metabolism of the cells?
That's a magnificent question. Indeed, the biggest part of the energy dissipation comes from chemical reactions. So when we are going to deal with, this is the last, I will have a last slide on that. When we are understanding, for instance, the metabolism, the glycolysis, and the Krebs cycle in the cell, the free energy changes are the order of magnitude per mole, like up to 60 kilojoules per mole, the change of the free energy. And the dissipation is going to be like 1% of that. That's what's going on. But so it's not, in most of the cases, the changes in the free energy are the most important. So it's just a fraction. The energy dissipation is just a fraction. But it seems sometimes, or in most of these active matter systems, we cannot explain what is going on just with the reversible changes of the free energy. We have to consider the energy dissipation that comes from the entropy production. And indeed, we can propose the non-equilibrium free energy that is also considering that an entropic part comes from the entropy change, irreversible entropy change. And that will be of importance and can be almost the same order of magnitude. I also going to show that in my case of the active pneumatics. But yeah, I understand sometimes it's kind of, for instance, when I started working on this, the order of magnitude of energy dissipation just because of the viscous dissipation is like 6 to 10 orders of magnitude less than the energy dissipation due to chemical reactions. And for instance, in a cell or living beings, chemical reactions is the main thing happening. So that's a very interesting question. So sometimes it's just a very tiny fraction, but sometimes when we go to these non-equilibrium free energy changes, it could be almost in the same order of magnitude. It becomes very important. Also because in the cells we are not in equilibrium. That's the point. Did I answer more or less?
Keep going.
Thank you. And then so we can find the energy dissipation for a given configurations of the tissue as a function, of the porosity and Young modulus. This is a map from our model. The point is that we know, or we found that there is a direct relation between porosity and Young modulus in the liver. So we can reduce all the analysis to one variable, that is the Young modulus, or as a function of the porosity, or as we want to do that. So maybe tomorrow I will be going more details on how to go from here to here. But we can compute from this map and our formalism, we can compute the probability currents in this space, because that's all about non-equilibrium, mesoscopic non-equilibrium thermodynamics. The most important variables are the probability densities and the current probabilities. So if we compute the current probability, each line or each frame corresponds to, for instance, the first one is for the healthy tissue, the second one is for the initial fibrosis, this green one is for the fibrosis stage, this white one is because there is nothing found for these values. This is the kind of the typical cancer for these values of the Young modulus, and then more stiff cancers. I don't know. The most bold cancers that are very hard to treat. The point is that the healthy tissue configuration is found to be in a minimum of the probability current, which means that it's very hard to see that the system is going to evolve toward this state. Whereas if we are in the carcinogenic, it's kind of everything is converging to this state, to this configuration. So it's at least an interpretation, and then we also find that as much, for instance, here, sorry, I forgot to mention, the order of magnitude of the energy dissipated in a tissue of, this was around 1 gram. We are in the order of the millijoules, which could be considerable, a huge quantity. But of course, it's because there is an external force also acting there. So from these current probabilities, we can also obtain the transition probabilities. The transition probabilities from the healthy to the initial fibrosis. As you see, it's almost nothing. And then and so on, and then we can find what is the probability to have this range of values that means cancer, and it's kind of it's less than 1%, 0.1%, which kind of makes sense. So this is another perspective to understand these tissues, the evolution of the carcinogenic tissues or cancer. Currently, we are working on another project. Hopefully, I will have time to explain a bit more in the last slide. That is very interesting because we also consider the metabolism. Now, the third part of my talk is about the active particles. Yes?
I'm just worried about units. So you said the energy dissipation is millijoules, but
Yeah.
you're in a steady state, so there should be a dissipation rate, or
No, yeah.
I missed something?
If we are in the steady state, of course, should be a flux, it should be millijoules per second. But here, what we were trying to understand was, if I have the tissue and I put an external force, and then we just compute the total entropy production through the relaxation process. Sorry, I'm going very fast.
That's okay. And about how long does that take?
Yes, so for this system, as far as I remember, it could be up to 30 minutes. Was the experimental data that we had access. The experiments were performing in Norway. So we validate our theoretical results with experimental data there. So now the third part of the talk is about the active particles. And again, it's to understand not only how the structures form, but why. Because these particles can be like an active gas, they can form something that is kind of orbitating, or sometimes it's collapsed. So we found 3 regimes. And also because, yeah, why active particles? This is the simplest case to understand some biological particles. That is kind of our main justification, or we started working on that because we wanted to understand the chemotaxis of some proteins, enzymes, some liposomes, some synthesized liposomes where in the surface my colleagues were able to put some enzymes to catalyze a given reaction. We work with catalyzing, with breaking the urea or breaking the glucose. So with two different enzymes, okay, so on. But the point is that there are two important things to measure here, and is the diffusivity, because it's an active diffusivity. It's not a passive particle, of course. And also when we have a gradient of a substrate or something, they are going to exhibit the chemotaxis that we also call it self-phoresis or self-diffusion phoresis, exactly, to be more exact, sorry. So the simplest case I mentioned is Janus catalytic particle. So essentially, it's in one part of the particle a chemical reaction takes place. So in the interface, because of this chemical reaction, the interface is going to experience some gradients, concentration gradients, also temperature gradients, because the reaction could be exothermic or endothermic. What else can I mention here? Oh yeah, we are going to have something that we call the surface excess energy that helps us to understand the active diffusivity. And here also will emerge surface tension gradients that is the origin of the phoretic forces at this scale. That is essentially like a Marangoni effect, but in this tiny scale that is going on in the interface of the particle, interface particle fluid. So we are going, if we consider these the gradients that are formed in the interface, and the surface tension gradient is the boundary condition for the hydrodynamic equations, we will have some streamlines like this. I hope everyone can see the streamlines, and they are not symmetric. There is a breaking of the symmetry, and that's the reason we can have the pushers or the pullers that are very popular in the literature now. And we also have here a gradient of concentration of a given chemical that is being catalyzed on the particle surface. And the first study case is for this vesicle. It's a liposome with some enzymes. So they carefully synthesize this liposome in which in the surface, just in one half, just in one phase, are most of the enzymes, which means that we are going to have an asymmetric particle that are going to be able to produce locally these gradients I'm talking about, these surface gradients, giving place to what we call the surface excess energy. So the particle is not under thermal fluctuations, so the thermal energy, but also is not under a noise that is not a white noise. That is how usually has been treated these particles in the literature. What we know is that they have a random orientation that follows a white noise, but the magnitude of this other force is the phoretic force, but is proportional to the surface excess energy. So we are able to compute this. It has different terms due to entropic changes, due to the thermophoresis, diffusion phoresis, electrophoresis, because they can also be charged, of course, in the medium. So not only the thermal fluctuation, the thermal force, but also this force will define the diffusivity of the particle, and is here. And from that, we are able to explain the experimental results of the diffusivity as a function of the reaction rate that the particles experience. So they were able to change this just by changing the concentration of in this case, the ATP, because was the hydrolysis of the ATP. But they can also play with this by changing the number of enzymes they can have in the surface. They obtain a non-monotonic behavior. So they weren't able to explain that at all. But then what I can explain is that there is a competition between the different terms of the surface excess energy that generates this non-monotonic behavior. And we were very happy with this, and we got accepted just a few days ago.
Sorry, what was the?
Yeah, non-monotonic as a function of the reaction rate, because you see.
What does the dashed line
no,
mean?
yeah. Sorry, yes.
What does the dashed line mean?
The dashed line was the first approach they proposed in the paper, because, as you said, because of the error bars are kind of big, It's kind of, okay, this dashed line kind of explain, but they were not very happy about that. And that's the reason we started working on something more elaborated. Sorry, can you repeat me, please?
It looks very suspicious that you have a curve that passes by the middle of points with huge Jagger bars. It looks like you are overfitting, like you have a potential that has too many parameters.
No, they are two main parameters in this case. One is the thickness of the
I understand.
of the layer, and the other parameter in this case was the derivative of the surface tension as a function of the temperature, because this is a very hard to find in the literature. So that was the other free parameter. So we had two free parameters to do the estimation, but there were only two.
But 2 parameters, I mean, to fit 4 points is not so impressive. But can I ask a general philosophy question? So I'm a bit, it's a bit different from what I'm used to one of these talks. So normally we have some open problems and we are trying to understand some open problems. Here, it's a bit different philosophy, right? So you are working with some experimental group in Norway, yes. They give you a bunch of points, and you are doing some engineering of trying to cook up some thermodynamic potential to fit those points and to fit, that's the spirit. But can you still put it in perspective within this big field of active matter? What problems did you manage to solve that were interesting and so on?
Yeah, so this was, I will try to answer him first. This is first part that I was mentioning. It was because we wanted to understand something that is an open problem is the chemotaxis or the chemotactic behavior of liposome vesicles and enzymes. This is a very open problem. And the first step to understand the chemotaxis is to be able to compute these coefficients, the active diffusivity and the phoretic coefficients that I'm going to talk later. So this is open. It has been, there are proposals from different perspectives, but the point is to understand why this matter, why these particles are moving with a given orientation or with a given, or they can have certain diffusivities that are larger than the passive counterparts. This is the open problem. I had my general theory and open questions. Then we have, yes, some applications that, but indeed, when I work with experimentalists, I have a chance to prove my theory. It's kind of how it was working in the past with the people in Norway and in Barcelona. I will continue the talk, and hopefully I will be able to show you the main view of my work. So to understand the chemotactic of this minimum cell, then we use Langevin equations. The phoretic force depends on what I was mentioning. The surface tension gradient. Here we are proving that exist chemotaxis is it means that these particles are migrating towards with the gradient. Again, we have big error bars because then the experiments, they were struggling a bit, but at least on average, they are not zero. And we are all able to see that experimentally and from the theory, because yes, since there is an excess of energy, of course, these particles are going to exhibit chemophoresis or chemotaxis, as you want to call them. Now I'm going to the collective
Can you
behavior.
comment a bit on what these experiments are?
Sorry?
What are these experiments? Why do they have big error bars? Why can't we decrease the error bars?
Yeah, sorry, I'm going very fast. This is for they were able to measure the velocity of the liposomes in a system in which we have a concentration gradient. In these systems, we also have Marangoni flows, that is the bulk, the bulk effect. So we have the drift because of the hydrodynamic force, but plus the hydrodynamic force, we have the phoretic force. So we were able to differentiate those forces and to estimate what is called, or what we call, the chemotactic velocity in a given direction. And that was, we also measured from the experiments. So and it was done for 2 cases, an enzyme that breaking urea, and an enzyme breaking glucose. And yeah, we were able at least to say, okay, they are migrating towards the gradient, and the coefficients of the chemotatic coefficient we estimated, we are able to present a value to explain this. Was kind of the spirit to show to the people, to the referees, that was accepted in a science journal. I already forgot. Yep.
Not liposomes. So
Yeah.
what is moving? What was the puzzle? Something was moving too fast, was moving too slow, something was not moving. Is it an animal? Is it a plant? Can you just explain in simple terms that people that do gravity waves or string theory can try to at least get an idea of what we are discussing?
So essentially, these liposomes are very similar to the vesicles. It's a membrane built from lipids. It's able to assemble, and it has a size of 100 nanometers. And in the surface, or in the membrane, better, we can set the enzymes. They can also be inside. So these enzymes are catalyzing a chemical reaction. So for instance, you can take ATP to break the ATP into ADP and phosphatum. We will observe gradients of these components in the surface, or here in the interface, also in the bulk. But the liposome is pretty close to some of the vesicles that we have in the cells. It's like an organelle. It's very tiny. It's very tiny. And the main thing why this was very important for the experimentalist is because it's kind of the simplest synthetic version of a cell, or the synthetic version of one of the organelles in the cell. And it also helped me to understand why of the movement of, for instance, a protein or an enzyme towards a substrate to react. This is very interesting in the field of, I will say in the field of biophysics, at least in chemical catalysis in this biochemical medium. So this is very interesting for the researchers in that field. Do you have other questions here? Nope. Then I will go to my original study, is to understand the.
One last one, and then I'll shut up. So
I'm sorry.
And then what do we do? Do we observe these guys moving? Do we have some way of tracking them?
Yes,
And we
yes.
draw, and then we study their average velocity as a response to some external stimuli?
The only external stimuli is that we set a constant concentration gradient. It's the only external stimulus.
Okay, so we are injecting liquid from one end?
Nope. Nope. They are. We have a system here. There is a concentration 1, there is a concentration 0. So we have a gradient in the concentration. Then we have in the dilute media, these liposomes, these small particles. So they are moving something like this. So in average, they are moving towards the higher concentration. We are able to track each of them in certain limits. At least we are able to track in the whole time of the experiment that can last till 30 seconds up to 1,000 liposomes. That's the reason the error bars it's good enough, the number, but we still have the error bars. But it's because the concentration gradient, we also have Marangoni effects that creates these fluid flows inside the chamber. But it's a closed system when there are the liposomes, and they will move in this direction, in average direction, because of the concentration gradient. Concentration gradient of the, could be ATP or glucose or urea. So it depends of which enzyme we are using in the vesicle, in the liposome, sorry. Yeah, sorry, I'm going quite fast, and I'm not mentioning a lot of details. I'm trying to. Okay, okay. So maybe I would like to stay here in the active particles for a while, because then here I will be able to present the. Everything started to understand. My goal is to understand the collective behavior of the active matter. So, as I presented in the beginning, I work in self-assembly and I have study cases, and then I wanted to understand the collective behavior of the active particles. The first step is to understand these very simple things that I just present. But of course, because we have a population of the particles, we are going to do statistics. And tomorrow I will go into detail of the formalism, that is the mesoscopic non-equilibrium thermodynamics. And the heart is that we propose the probability conservation in the space where we are describing our system, what is going on in the system, and the entropy, and the Gibbs entropy that I mentioned briefly before. We can obtain the entropy production rate, that is the thermodynamic force times the probability current, and with the Onsager formalism, we can write the probability current as a function of the thermodynamic force, and if we insert this current into the probability conservation, that this is the probability current, we can have like Fokker-Planck equations. So let's see to understand the collective behavior, from Fokker-Planck we can also obtain Langevin equations, the macroscopic equations for the concentration field. For instance, if we are using the ATP as the fuel, and also we are able to obtain the hydrodynamics equation. So this is the first main thing that I'm showing here, that is from this thermodynamic formalism, we can obtain the dynamics of my system. All the dynamics, the translation of the particle, the rotation of the particle are coupled, and they are also coupled with the other fields, like the concentration field or the hydrodynamic field. So that's kind of the key idea here, is to show, mainly for the community in active matter, that the coupling between the movement of the particles and the surroundings is important, because they are forming because they are changing the surroundings, and vice versa. When the surroundings change, the possible structures that we can have also might change. So this is something, it was an open question in the literature because the models are very simple. This is a snapshot of what we can have in the simulations when solving the equations. The fluid field, the concentration field, and how the particles start to interact to form some clusters or not, because they can behave like an active gas, or they can start to orbitating among themselves. And for instance, we have a system, we have two possible systems. One where the initial condition is that we set a droplet of the chemical fuel, could be ATP. And so the particles are going to migrate, and they're going to move faster, and they can form different structures. And then we have other system. This system is going to be close and isolated. We are not going to talk here about production of heat. And then there is other system where the initial condition is homogeneous, but at the end of the day, we are going to have essentially the same regimes. We are going to observe the same structures when we use the catalytic Janus particles. This is, for instance, how they are evolving on time. For instance, in this case, they are kind of not interacting, and then they started to interact, and then here, for instance, they are forming a cluster. It's kind of, that's called a chemotactic collapse regime. So usually people in working on active particles do this, and they create amazing maps with different possible regimes and structures. So we wanted to recreate that with the main coefficients I was talking before, this active diffusivity and the chemophoretic coefficient. And for these different two parameters, we obtained the different structures that is observed in the literature and also in nature. For instance, in nature, to assemble some of vesicles, we will be interested on something like this, or to see how some proteins aggregate is also something like this. So this is the kind of the interesting thing that we wanted to observe. Of course, for each point of this heat map, we can also compute the total entropy production. From the total entropy production, I mentioned before that with thermodynamics, we also kind of propose some principles, or we can delimitate the why some structures are being formed or not. In this case, our parameter to understand the system is the fraction of assembled particles, and is the number of particles that are being part of a nucleus over the total number of particles. This is just the fraction of particles, and this is indeed the structure here mathematically is essentially pretty similar to classical thermodynamics. So we can compute the total entropy produced in a time window, so it's not going to be a rate. I'm going to talk about the total entropy produced for the formation of the structure. Then after a while, the structure are going to disappear. Why? Because if there is not more chemical fuel, there could be the ATP or glucose. If there is not enough, of course, they are going to be just under the thermal noise, and they are going to disappear. So the important thing that I wanted to mention is that we can propose this kind of non-equilibrium chemical potential that depends on the reversible counterparts, but also depends on the entropy production, the total entropy production. And then this change of the chemical potential is very important, because when it's negative is when we observe the regime that is dynamic self-assembling, that are the particles that are orbitating among themselves. And then the chemotactic collapse that is the most found in nature is this part. So, and it's negative, which means that spontaneously the structure assembled, because energetically it's favored from the thermodynamic point of view.
Sorry. Oh. No, it's just a quick question. So how much of this phase diagram and all these thermodynamic properties depend on the model of active particle that you choose? Because you chose Janus particles.
Yes.
But I mean, there are many others.
Yes.
So you chose Janus particles because they are closer to your biological system, I guess.
Yes.
But is all this phenomenon robust to other type of active particles? So it's specific of Janus particles, and how all this fits within active matter more broadly?
That's a very interesting question. The nature of the discrete unit is crucial. So, for instance, if we have elongated particles, this could change a bit. In the regimes that we can observe is not going to be, perhaps they are not going to be these ones. Maybe we are going to have another regime. What I'm trying to say, this is not general. This is for the Janus particles.
So this dynamic self-assembling is similar to, now I forgot the word, MIPS.
Yes,
okay.
This induced mobility. Yeah, the
mobility
MIPS.
induced phase separation.
Yeah. Yes, exactly. This is. So what we were more interested was in this part, because it's closer to what we find in biological matter. Yep.
Clarification, what are the dots?
The dots.
No, in the next slide.
In the
You have
next.
dots and solid curves. Can you? What are the dots?
Oh yeah, this is we are taking here around this value. Of course, we were not able to do all, or to take, how to say it, to run all the values. So we are taking like the 0.5, 0.1, 0.2, and so on. Strictly, we will have some error bars, but yeah, because of the computational limitations, we are not able to explore all the points that we were aiming. So each point is the point around we were performing the simulations. So because each simulation for each point was taking a lot of time. So that was our limitation in that case. And then the continuous line is a tendency. Yeah, that was a limitation, a computational limitation to have more points, yes. Okay, now I'm working with active nematics, and to describe the system of active nematics, we have three components. The nematics itself, the substrate, that as I mentioned could be the ATP, and the fluid molecules. So to characterize the nematic, they have a position, an orientation, the chemical reaction that I will talk generally of active nematics, but for instance, in the case of microtubules, that we have these complexes with the myosine systems, if we do a zoom, they essentially are moving kind of in this direction and this direction. So I'm going to simplify that to understand that is like this an excess energy, or excess not necessarily, this is a surface energy. So because of the reaction and the movement of the motors, there is more energy at the surface. That's it. This is the simplification. So microtubules are these long units that are fundamental to give the structure for the cells. They are very important in the cellular division. They kind of, yeah, I wouldn't say dry, but it gives the structure for the cells, for instance. So now in active matter community, in the ones working with active nematics, understanding the movement of the nematics and the creation of these topological defects, that this is another concept that is used in the field, it helps to understand the self-organization of the microtubules and the migration of the microtubules that is of the high importance in biological process, some biological process. So the point is that if we can characterize the system with the components, with the main variables, again tomorrow I will go into detail of this. We can obtain we apply the formalism, non-equilibrium thermodynamics formalism. We can obtain the nematodynamics. So it's just the equation of the dynamics and evolution of the Q tensor, that is the nematic tensor, and from that we can compute the order parameter to find the defects. So the main thing here is to be able not only to explain what is already very well known by the community in active nematics, that is the nematodynamics, that is this one that is in blue, but from the thermodynamic formalism, we can have more terms, extra terms accounting for other forces that are crucial to understand, for instance, here for the nematic concentration, to understand why of the migration of the nematics, because they are by far not homogeneous in the system. And then this is what I'm going to show in the our motivation for the project that I'm currently working on. Here also we are able to propose the concentration field, the fluid dynamics, and the active stress tensors. Indeed, this is a generalization of the active stress tensor because they are right now working with a good approximation, but they consider that the activity is just a coefficient that what means physically that coefficient. So for me, this coefficient depends on the surface excess energy that at the same time is depending on the chemical kinetics and on the coupling coefficient that emerges naturally from the Onsager formalism. So activity is that, is not only, I don't know, as I mentioned at the beginning, activity is defined in active matter like it will depend. It's not the same the activity in a Janus particle that the activity in the collective motion of birds or fish, no. So that's the physical interpretation that we can give to activity in active nematics, and that's the reason I started working with people there in Ljubljana, in Slovenia. So this is a very interesting paper where for a given confinement, there's a system with size 200 micrometers with the microtubules. They put the molecular motors, the myosin, and they start observing the flows and the variation in the concentration of the nematics, and as a function of time, the defects, the topological defects, they are not always the same. They are varying, oscillating on time. So we wanted first to explain this effect here of the density of the nematics, and also we were interested on the evolution of the topological defects. So this is the experiments, how the system is evolving on time. As you see, most of the microtubules are now almost in the boundaries, and the number of defects are kind of changing as a function of time. So they have multiple configurations. Is there an energetic explanation? Because in this field of nematics, and in active nematics, they are aware that not all the configurations corresponds to a minimum of the free energy of this Landau, the Jens free energy that they work. So they want me to explore from this dissipative point of view the formation of the different configurations, and if we can explain why they are varying on time, we can have 2 defects or 4 defects, and why they are having this migration of the microtubules, because up to now there are not models doing that. So what I started doing, sorry, I don't know. Yes, it's evolving now. So this is some preliminary results where these blue points corresponds to the defects. As you just saw, they are oscillating between 2 and 4. I don't know if we can see very well, but this is the fluid field, the velocity. And in this is the evolution of the concentration of the nematics. Of course, this is a continuum approach. We can also go to the discrete, but for now, we kept in the continuum one. Here the variation is up to the 50%. The concentration here is almost half as the one found in the boundaries, which corresponds to the experiments, as well as the fluctuations here. So we are very happy about that. I also wanted to show them that actually we will have temperature gradients, or average temperature gradients along the system, because usually we assume that the temperature is constant, but it is not necessarily constant. And here, I wanted to come here. The different contributions for the entropy production rate. So we have the viscoelastic contributions because of the viscous and elastic nature of the fluid. We have irreversibilities, then entropy production, that is this term. Because of the active tensor, we have another contribution that is order of magnitudes lower than the entropy production because of the chemical reactions and because of the diffusion. So the most dissipative processes that we have in nature are the diffusion process, and the chemical reactions. And most of the times is the chemical reactions. For that reason, it's super important in active matter, in active living matter, to consider the reactions and the free energy changes on the reactions, because can dominate. Then this is a new term that I found that is because of the diffusion of the nematics that is very important in this case too. And this is another classical term that is the dissipation due to the degree of freedoms in the orientation of the nematics. So basically the dominant one is the reaction and this diffusion term. We can propose an effective chemical potential.
Sorry,
because
can you just explain the notation? Just, I mean, maybe it's something very, very simple. Just the previous slide. So del is a vector. I just want to understand what that double bar squared thing means.
Yeah, this.
Yeah.
Okay, this is the norm of the concentration gradient of the substrate in the system. Yeah. This is the diffusivity of the.
So the double bar just means that you're just taking the dot product of the vectors. Right?
Yeah.
Okay, fine. And on the second term.
Yeah,
P
yeah.
is a scalar.
Yeah, it's going to be the same.
P is a scalar. Where's the other vector? Just to understand. What's the vector in the second term?
This is the vector, and
in the first term, I understand it's a vector. Where's the vector in the second term?
This is a tensor.
Ah, Q is a tensor. Okay.
This is a tensor.
Okay,
that's the reason. Yes.
Okay, thanks.
And then I just was a bit lazy. Sorry. So yeah, I was going here.
You're on the next slide.
Yeah. So a bit of thermodynamics is just energy balance. No, this is not just an energy balance. This is a free energy balance. First, I want to understand the free energy variation of the system, because since we are in a kind of a steady state system, there is a rate of change in the free energy. There is a rate of change of the reversible free energy that comes from the elastic nature of the system. Because we have nematics, then I don't know if you are familiar with this Landau-de Gennes free energy to describe these systems. So whatever, this is classical. And then we have the energy dissipated. And then, of course, we have this is something that people usually forget. We have a chemical reaction. The chemical reaction has a change in the free energy between products and reactants. And this is the input of the energy of the system. So the system is active because actually the chemical reaction is giving the energy to the processes that happen. That's the key point. But yeah, the point is that I wanted to see or to define a kind of potential that this is very classical. This is how the free energy varies with the amount of molecules, let's say. If we are in a steady state, let me do this. It's weird, but it kind of more or less makes sense. So we can have the variation of the free energy, total free energy, as a function of the free energy input that is because of the chemical reaction. So this is pretty similar to a chemical potential, just that the temperature, neither the pressure is constant at all, so this is something effective. But it kind of has the same structure of the chemical potential in classical thermodynamics. And it will depend on the dissipation, of course, depends on dissipation. So by far, this is the most important result that I have obtained from this theoretical point of view, and is that the second derivative of the energy dissipation as a function of the reaction rate, let's say, I'm plotting here the dissipation rate as a function of the free energy rate from the reaction. I don't know how you see the colors here, but these things that we are seeing here is the number of topological defects that is fluctuating from 2 and 4. And this function is the dissipated energy. So every time the system relaxes towards two defects, that is kind of the, let's say, the basal state, it reaches a maximum in the energy dissipated. So something with the second derivative equal to 0 is giving us information when and why the system is relaxing to the basal state. This is another result that is not general at all. I did this for different configurations. And yeah, but this is the only thing that I found that is by far general. I was expecting that around the maximum of the energy dissipated, something was going to happen because it's what I proposed since my PhD. But in pneumatics, I think he's gone. In pneumatics, for instance, we are not able to have the same regimes or behavior as in active particles, which makes sense.
So are you concluding soon? Because you're already over time.
Oh, God. Yeah, I'm sorry. I just want to mention that I'm working in cell sorting. This is very interesting because we have the non-reciprocal interactions. We are able to describe that with mesoscopic non-equilibrium thermodynamics. We add another equation for if we want to describe in Langevin space, and then we can also have the macroscopic version, which is very interesting because it's the typical, is this typical reaction, diffusion that are able to display these spatiotemporal patterns. The final slide is another project that I'm working. We are still waiting for some money. Is the cancer prognosis, and it's now considering the metabolism and the role of the transporters, is these proteins or these channels that we can also nicely describe with our formalism. And here we are talking about something more realistic. So we are working here with people from in Poland and this Institute of Cancer there in Varsovia. I forgot the name in English. And few conclusions. Energy dissipation drives the emergence of self-assembled, self-organized structures. The non-equilibrium potentials allow us to explain the spontaneous formation of structures in active particles and active pneumatics. The structures both depend on and modify their environment. It's not biseparated. That is what is usually done in literature. And order emerges and evolves because the system is transforming the energy at the specific levels of dissipation. So it means that they are close to a given maximum or minimum dissipation. And final question is the formation of biological form, the regulated heat production that could be related to thermogenesis, and the emergence of life itself reflect a shared physical mechanism. It means by which active matter organizes under sustained energy and flow dissipation, which means can we find this general law that allow us to understand not only active particle or active pneumatic, but also what I was mentioning with the liposomes, with the proteins, with the formation and cellular division. And this is the big question, and that I have been working in smaller projects to try to answer this, and I am still trying to answer that. Thank you very much for your attention and questions.
Okay, thank you. Questions?
Yeah.
You could say this is not the right time to ask this, but I have a kind of philosophical question. So the problems in active matter are motivated by the problems in real living systems.
Yes.
So let's pick the sort of chemotactic behavior of your liposomes. Do I suppose, okay, let's fast forward the research program, you learn everything, you completely solve that problem. Do you feel like you're learning something that would inform our thinking about chemotaxis in bacteria?
Yes, absolutely.
Okay, so can you give a short summary of what you imagine that would look like? Or, because I mean, one of the problems that I'm having with this is that, you know, people physicists have been studying chemotaxis in bacteria for 50 years, let alone the biologists. So, and by now the error bars are smaller. So, so I'm trying to figure out, if you're really interested in chemotaxis, maybe you should just study bacteria. So tell me, what's the thing that I'm going to get by doing this?
Yeah, for instance, something closer should be the case of the, this is cell sorting, but to understand self-sorting, first we must understand the movement of the cells by itself, themselves. Here we have two kinds of cells that after interaction, they will have different behaviors, whatever. The key point here is that the dynamic, the chemotaxis of bacteria, I will take this one. They are elongated, so indeed there are people that are in the institute working as they are nematics because sometimes they exhibit nematic behavior, blah, blah, blah. But I'm more interested in the chemotaxic behavior. So they are going to have they are going to orientate towards a chemical gradient. This is chemotaxis. The point is that I'm always saying that the process, the phoretic force that allows that, they are a given position. They are going to move towards a given direction because they have a phoretic force. Sorry for you. They have a phoretic force. And they also have an orientation. So the orientation kind of should be fixed. Otherwise, the bacteria is going to move like this. And we want the bacteria to do, at least on average, to do something at least like this. It's migrating in a preferred direction. So since they have different mechanisms, indeed, I was in a conference where there are like 56 different taxes. Anyway, they have different mechanisms, mechanical mechanisms. But if we try to simplify a bit from the thermodynamic point of view, we can understand this here. For instance, if they have the flagellum, is like an excess in the surface energy, and then this excess of the surface energy can also help us to compute the phoretic force. And since they are sensing, because they have different proteins, they can sense, and there is an internal mechanism. They can sense the gradient. We can establish. Indeed, I didn't talk here because, as you say, I had a lot of things. We have a rotation potential that give us a good results in active particles and in other particles to understand thermophoresis, for instance. So we can also define the orientation dynamics like that. And since all this mechanism is also related to a polarization in the membrane, if we consider another general parameter, we have another Langevin dynamics that is like a polarization, reaction-like equation, blah, blah, blah. I have no name for that because I'm still working. We could understand the movement of this object or the bacteria and to understand the chemotaxis of the bacteria. So I will expect something like that, something similar to what I have done with the active particles and what I'm doing now with these cells. So far, yes.
I'm probably very naive, but I'm still a bit confused by this general philosophy because you find these potentials which I should think of as some kind of phenomenological potential, right? To describe either this rotation or whatever, right? Or entropy production and so on. Then you have some curve for this potential, which is something complicated. And then if you do something biological, probably the potential is a bit different. And different materials, there will be different potentials.
Yes.
So what exactly are we learning? It looks like I just see some curve. Okay, fine, I fit some points, but who cares? Then I change the material, I get a different curve.
Yeah.
Why is this interesting?
No, no, absolutely. But indeed, this is the main point. What I want to say is there is a definition of the potential of the effective potential that depends on the reversible part, another part that depends on the total entropy produced, and another part that also depends on the dissipation. This is something that we found, and it will describe different processes. But you are right, depending on which variable we are using here, depending on the material, depending on the objects, depending on the constraints, we will have a curve like this or a curve like that. I don't know, but this is a potential. Also, for instance, when we have a chemical reaction, that tomorrow I'm going to talk about that, we also have a potential along the reaction coordinate to go from A to B. And in general, we know that it's going to be something like this. But for the reaction A giving C, it could be something like this. And yeah, it's going to depend on the material, the process.
So then what do we learn? Because I assume that what we want to learn are about mechanisms, right? Why things work, what type of,
Okay.
why do these particles move the way they move, why do they turn more in one direction or another? If I look at the potential, okay, I look at the potential, but
Yeah.
from the potential, do I learn things about the underlying mechanisms? What's the, it's like statistical mechanics and thermodynamics, right?
Yeah. So from this formalism that also consider the potentials, we not only want to understand the mechanisms, we also want to understand the energetics. From the potentials and from what I was showing, this behavior of the chemical potential as a function of the fraction of assembled particles, what I want to learn, indeed, thank you very much for your question. Now I remember that I forgot to mention something very important, and it's because these curves here, they are very far away. No, actually, no, it's my fault. It was so important and I forgot to mention. Of course, in this case, the maximum, what is going to correspond the maximum, or at least what we found that is the maximum of the entropy production, is where we observe the transition between the active gas behavior to this dynamic self-assembled behavior. There is this state where they are rotating kind of around themselves. And this other maxima corresponds to the other transition between that regime to the chemotactic collapse regime. So these points along the last 100 years in thermodynamics to understanding what means the maximum entropy production principle or the minimum entropy production principle. From the thermodynamic point of view, this is key and is not understood at all. So there is always a question is respect to what? For instance, is maximum respect to what? So something that I try to respond is, okay, is respect this structural parameter, the maximum are giving us information about when we find transition in the systems. When we go to the potential, the zeros corresponds to the transitions. And negative values of this potential corresponds to the energetically favored structures, which means that that's the reason here we observe this collective behavior, and that's the reason here we observe the chemotactic collapse. That is what we observe in biology. So what I'm trying to say is that this agglomeration from the thermodynamic point of view makes sense. Energetically, they are favored. So this is the why. Why the structure is formed? Is formed because the chemical potential difference is negative, which is exactly equal to what we do in classical thermodynamics and classical statistical mechanics when we go to use the chemical potentials, for instance.
It's like saying the box shrinks because pressure is negative. Okay, it's not understanding. It's just translating.
Okay.
things into a potential. Okay, if you draw the potential and see where the energy is positive is negative, okay, fine. It will favor this region and not this region. I would not call that understanding. It's describing.
Okay.
You are describing which regions are important or not, based on simulations, based on data, based on fits, but you are not understanding.
Yeah, because the understanding from a mechanical point of view is to explain the mechanism, why they are being formed. Okay, this is the how. This is the how for me. In the dynamics, I can say, or I can try to explain from this point of view is that when we have the chemostatic collapse, or the particles are very packed, it's kind of the particles that are in the center, they are losing energy, so they are kind of reaching more basal state. But the ones that are here in the, let's say in that interface, they are first reproducing the maximum entropy production in the system, just here in this interface. And then what's happening is that they are trying to migrate towards the minimum, in this case, towards the minimum concentration of the substrate, because then they are kind of, they can be understood like they are trapped or they are more stable. So again, yeah, maybe because I have worked a lot with thermodynamics and with the energetic perspective, here this is energetically favored because they are reaching lower energies by sacrificing, let's say, that here in the interface, they reach the highest entropy, the highest values of entropy production. So mechanisms are it's hard to explain. Yes, when you have a gradient. Yeah, and yeah, it's because the mechanism it has a lot of details. With thermodynamics, we always synthesize. That's the point.
Any other questions? Okay, so Andre will give another talk tomorrow. Thanks so much.
Thank you.